Welcome to episode 21 of Pulse of Physiology. I'm your host, Dr. Jim Davis, and today we'll uncover the complexities of vision from how light activates photoreceptors to the processes of light and dark adaptation. The human visual system is a marvel of biology, converting light waves into electrical signals that the brain interprets as the vibrant world around us. In our next episode, we'll delve into the functions and control of the endocrine system, but for now, let's focus on vision. So to start off, let's discuss the pathway of light. Light enters the eye and follows a specific pathway to enable vision. Light passes through the cornea and pupil where it is focused. From there, it goes to the lens, and the lens fine-tunes the focus of light onto the retina. The retina is where light activates photoreceptors, and this initiates the process of phototransduction. Now, within the retina, there are three main types of cells. You have photoreceptors, which are made up of rods and cones. Rods are sensitive to low light and located on the periphery of the retina. They are crucial for night vision but do not detect color. Then there are cones. These photoreceptors are activated in bright light and are concentrated in the center of the retina, also known as the fovea, and they are responsible for color vision and high acuity. In addition to photoreceptors, we also have bipolar cells. These cells act as intermediaries between photoreceptors and ganglion cells. And there are two different types of bipolar cells. You have on bipolar cells and off bipolar cells. Now, on bipolar cells depolarize in response to decreased glutamate release, which occurs when there is light. Off bipolar cells, on the other hand, depolarize in response to increased glutamate release, which occurs in the dark. Glutamate release by photoreceptors inhibits most on bipolar cells in the dark, preventing the transmission of signals to ganglion cells. And ganglion cells are the third type of cells in the retina. And ultimately, these cells generate action potentials where signals are transmitted to the brain via the optic nerve. So we're going to take these three different types of cells and ultimately go through phototransduction, which is how light becomes an electrical signal. Now, phototransduction begins when light hits the retina, causing chemical changes in photoreceptors. In the dark, rhodopsin, which is found in rods, is inactive, and cGMP keeps sodium channels open, which depolarizes the cell. These depolarized photoreceptors release glutamate, which, as I mentioned a moment ago, inhibits most on bipolar cells, and as a result, the brain perceives no light. Now, when there is light, light activates rhodopsin, and this separates into retinol and opsin. And when this happens, we refer to opsin as being bleached. Now, activated opsin stimulates transducin, which is a G protein, which activates the enzyme phosphodiesterase, abbreviated as PDE. Now, PDE breaks down cGMP, which closes sodium ion channels and hyperpolarizes photoreceptors. Glutamate release then decreases, which allows on bipolar cells to depolarize and excite ganglion cells. Action potentials are then sent to the brain, which signals the presence of light. So let's talk about color vision and cones. Now, there are three types of cones, each sensitive to specific wavelengths of light. You have blue cones, which are activated by short wavelengths, green cones, which are activated by medium wavelengths, and red cones, which are activated by long wavelengths. Now, color perception is based on the combination of cones activated by specific light wavelengths. For example, a wavelength of 500 nanometers might activate 20% blue cones, 80% green cones, and 50% red cones, producing the perception of a specific color. Now, light and dark adaptation are also important when it comes to vision. These adaptations allow the visual system to adjust to changes in light intensity through light adaptation and dark adaptation. Now, light adaptation occurs when we go from the dark to the light. Rods and cones become highly sensitive to light and darkness, meaning a little bit of light will activate these photoreceptors. However, when we go from dark to light, the bright light causes rods to deactivate, and the cones are overwhelmed by the amount of light. However, over the course of about 60 seconds, the cones desensitize, which allows your visual acuity and color vision to improve. Then there is dark adaptation. Dark adaptation occurs when we go from the light to the dark. In bright light, rods and cones lose their sensitivity, meaning it takes a lot of light to activate them. However, rods become bleached because there is so much light, and so they're inactive. In the darkness, rhodopsin generates and retinal sensitivity increases over 20 to 30 minutes, restoring night vision. In other words, the photoreceptors are becoming accustomed to only having a small amount of light to activate them. Now, understanding these vision mechanisms can help diagnose and treat visual disorders. For example, there's night blindness, which is often caused by vitamin A deficiency, which affects the ability of rhodopsin to regenerate. Color blindness is a genetic condition where one or more cone types are missing or non-functional. And then there are retinal disorders, so conditions like retinitis pigmentosa damage photoreceptors, which impairs vision. So let's summarize with our key takeaways. Vision begins when light enters the eye and activates photoreceptors in the retina. Rods are crucial for low light vision, while cones enable color vision in high acuity and bright light. Phototransduction converts light into electrical signals through the actions of rhodopsin, transducin, and PDE. On and off bipolar cells respond differently to glutamate, enabling precise signaling in light and dark conditions. Light and dark adaptation ensure optimal vision across different lighting conditions. And so that's it for episode 27 of Pulse of Physiology. I hope this exploration of vision has deepened your appreciation for the complexity of the visual system. Join me next time for episode 22, where we'll discuss the functions and controls of the endocrine system. Until then, keep questioning, keep exploring, and stay curious.